Today we are talking about a wonderful gram-negative coccobacillus bacterium by the name of Legionella pneumophila.
Legionella is fastidious, meaning that it needs a lot of nutrients to survive. It is also anaerobic with a single flagellum and lives within soil and freshwater amoeba.
This bacteria is most well known to cause a type of fatal pneumonia called Legionnaire's disease. Let's learn more about it.
Did you know that Legionella pneumophila evolves by infecting protozoa within freshwater reservoirs?
No, I did not, but that's really neat. Well, how does the host become infected by Legionella pneumophila?
It is spread by aerosolized droplets and breathed into the host's lungs. It then comes into contact with the alveolar macrophages once it has been ingested. Here it is able to replicate after it changes the harsh environment to a more hospitable one inside the macrophage.
So how is the bacteria able to survive the harsh environment inside the macrophage?
If Legionella bacteria has the gene./ICM, it is able to create a Legionella-containing vacuole, or LCV. It creates a double membrane vacuole that is formed by the help of the host endoplasmic reticulum. The bacteria also has several other virulence factors that contribute to its overall pathogenesis.
What are some of the virulence factors present in Legionella?
For one, Legionella is able to prevent phagosome acidification and phagosome lysosome fusion by recruiting host protein RAB1 to the phagosome. By recruiting this protein to the phagosome, the pathogen is modifying the identity of the phagosome. In total, Legionella secretes around 300 effector proteins to manipulate the host environment.
These Legionella pneumophila containing phagocytes are isolated from the endosomal pathway, embodied within the endoplasmic reticulum, creating an intracellular niche that is favorable for the bacteria to replicate. When the vacuole can no longer support replication, a regulatory cascade coordinates entry into stationary phase, promoting the transmission of the bacteria to a new host cell. In this way, Legionella pneumophila is able to cycle between an infection replicative form and non-replicating form, thought to promote transmission to a new host cell.
Legionella is also able to suppress UPR, or unfolded protein response, blocking the translation of UPR genes and proteins. It is able to block UPR in multiple ways, one being the splicing of regulators of UPR.
RPSL protein is responsible for the induction of host cell death through the activation of caspase-3. Caspase-3 ultimately leads to loss of lysosomal membrane integrity, releasing hydrolases into the cytosol, resulting in its processing of substrates and cell death.
There are also virulence factors associated with the cellular envelope of Legionella, including outer membrane vesicles, or OMVs for short. These fuse to eukaryotic host cells, bringing in virulence proteins and factors, and ultimately inhibiting the joining of phagocytes and lysosomes. This prevents the host cell from killing the Legionella.
How was Legionella detected by the host?
The host utilizes TLRs, or toll-like receptors, to stop Legionella pneumophila. Toll-like receptors are proteins that play a major role in the innate immune system. They are key in recognizing pathogen-associated molecular patterns, or PAMPs, and ultimately defending against invading pathogens. In regard to Legionella, the host cell membrane utilizes TLR2, TLR4, and TLR5 to stop the intruder.
So what role does each TLR play in recognizing the bacteria?
In short, TLR2 functions in detecting lipoproteins of Legionella and recruiting chemokines to signal to the rest of the cell that there is an intruder present. TLR4 recognizes lipopolysaccharides, and TLR5 is able to detect the presence of singular flagellum of the Legionella pneumophila.
Oh, don't forget the other TLR involved in defending against Legionella. One additional TLR that plays a role in detecting this pathogen is TLR9. Unlike TLR2, TLR4, and TLR5, TLR9 is found within the intracellular phagosomal department rather than the cell membrane.
Well, what is the function of TLR9?
TLR9 is able to detect unmethylated CpG DNA, which differs from human DNA in that it becomes methylated later after replication. In short, TLR9 lets the body know that this CpG DNA is non-self.
The final innate response used to defend against Legionella pneumophila is recognition in the cytoplasm by seven different receptors. While I will not go into extreme detail, cytoplasmic receptors are split into subgroups with different functions, including recognition of bacterial and viral nucleic acids through PAMP recognition, similar to TLRs, activating transcription factors in order to mediate expression of inflammatory response genes.
Cytokines also play a role in controlling growth. Cytokines are released by cells surrounding those that have become infected. Cytokines that are released are pro-inflammatory cytokines.
How does inflammation help? Normal cells that usually form a tight junction by creating a wall between each other will now become leaky and allow for innate immune cells to access the site of infection and clear it. The cytokines play a key role in circumventing the blocking of protein synthesis by Legionella pneumophila, which without them it would be extremely difficult to recover from this bacterial infection.